All right, let's talk about something we think is simple, cleaning. But we're going way past just wiping down a counter. We're diving into the science of sterilization, which is the process of making absolutely sure something is free of all living microbes. It's a high-stakes world where being almost clean just doesn't cut it.
So, let's kick things off with a big question for you. Think about it. We can blast a metal scalpel with heat to sterilize it. Easy. But what about all the stuff that heat would just flat out destroy? How do you get something perfectly, medically sterile when you can't just torch it?
And that right there is the huge problem. Using heat, especially moist heat, is great for some things, but it has some serious limitations. It's basically useless for anything oily or powdery. And, more importantly, it's a complete non-starter for heat-sensitive stuff, like a lot of modern plastics or delicate medicines that would just be ruined by high temperatures. We needed a whole new playbook.
And that new play is filtration. You know, the concept is actually beautiful in its simplicity. If you can't kill the germs with heat, well, you just physically pull them out. It's like a microscopic colander. You pour your liquid medicine through it, the good stuff passes, and all the nasty bacteria get left behind.
Now, this idea has been around for a while, and the early versions were quirky. You had the Sites filter, which used asbestos pads. Yeah, not exactly a long-term solution, right? Then you had sintered glass filters. They were better because they didn't soak up the medicine, but they were super fragile and expensive. And the old ceramic filter candles, they worked, but they clogged up all the time. These were okay starting points, but we really needed something better. Which brings us to the hero of our story, the modern membrane filter. This is a tech that completely revolutionized how we sterilize sensitive products, especially things for parenteral use, which is just a fancy way of saying anything that gets injected or infused right into your body.
So what makes these filters the undisputed champs? Well, first, it's just straight-up physics. The pores are so uniformly tiny that bacteria literally can't squeeze through. This structure also means you can filter things incredibly fast. They're also disposable, which is a total game-changer for stopping contamination between different batches. And maybe best of all, very little of the actual medicine sticks to the filter itself, so you don't lose your valuable product. It's the whole package: speed, safety, and efficiency.
And it all comes down to this one magical number, 0.22 micrometers. That's the standard pore size. To give you some perspective, that is mind-bogglingly small, almost 400 times smaller than the width of a single human hair. It's that incredibly tiny size that lets us physically block bacteria, guaranteeing that what comes out the other side is truly sterile.
So, the process is running, the sterile liquid is being collected, we're good to go, right? Well, not so fast. This is where the science gets even more intense. How do you know for sure? How can you be 100% certain that every single microbe was caught? How do you prove it?
Because in this world, you can't just hope it worked. To put that sterile label on a product, you need cold, hard, verifiable proof. It is absolutely non-negotiable. And this is where sterilization indicators enter the picture. They're the tools we use to watch over the process and confirm it was a success.
I like to think of them as our sterility detectives. They're like a quality control team, and they investigate on 3 separate levels to give us the full story and prove, without a doubt, that the sterilization process did its job.
And these 3 levels build on top of each other. We start with the physical machinery checks. Then, we bring in our chemical witnesses that give us a clear visual sign. And for the grand finale, we have the ultimate test, biological proof. Each step adds another critical layer of certainty.
Okay, level 1 is all physical. This is about monitoring the equipment itself. Did it run for the right amount of time? Was the pressure correct? Think of it like checking the dials on your oven. For filtration, a super important physical check is the bubble point pressure test. You do it after the filtration is done to make sure the filter membrane is still perfectly intact and didn't get a microscopic tear during the process.
Next up, level 2, are chemical witnesses. A classic example is something called a Brown's tube. It's this little glass tube filled with a special red liquid. When that tube is exposed to exactly the right temperature for exactly the right amount of time, a chemical reaction happens, and poof, the liquid turns from red to green. It's a simple, undeniable yes or no. If it's not green, the batch is a failure, period.
But even that visual proof isn't the absolute final word. For the ultimate confirmation, we have to go biological. And this quote here, it says it all. It measures the process directly by integrating all the different factors at once.
So what is this final ultimate test? It's the gold standard. You literally challenge the system with the toughest opponent you can find. You take a sample of incredibly resistant but totally harmless bacterial spores. Think of them as the special forces of the microbial world. You put them through the same sterilization cycle. Afterwards, you try to get them to grow in a lab. If nothing grows, if they're completely dead, then you have definitive proof that your process is powerful enough to kill even the toughest bugs out there. It is the final boss of sterility testing.
So, we've talked about filters, tiny pores, and 3 different layers of proof. Why on earth do we go to all this trouble? Well, the answer brings all this science right from the lab bench to your bedside.
And this, right here, is why it matters so much. Every time you get an injection, use sterile eye drops, or get an IV in a hospital, you are being protected by this incredibly rigorous, multi-layered system of quality control. It's this hidden science working completely behind the scenes that makes modern healthcare not just possible, but safe for all of us. And that really makes you think, doesn't it? This whole complex world of sterilization is just one of these amazing, invisible processes quietly working to keep us safe. So I'll leave you with a final question to think about. What other invisible processes protect you every single day?